What are chromosomal aberrations? Structural changes in chromosomes are called chromosomal aberrations. Such changes do occur in plants and animals, including man. Due to these, behavior of chromosomes is highly affected during meiosis. Structural changes lead to behavioral changes in the chromosomes. Pairing between homologues of crossing over is obliterated, and it may result into unviable segregants. All four types of gametes in relation to the genes involved may not be formed as in case of normal chromosomes. If a cell or organism has a pair of homologous chromosomes with one normal but its homologue being aberrant, the condition will be called as heterozygous. Such cells or organism will be referred as heterozygote. Here these terms are in relation to chromosomes that vary structurally. Chromosomal aberrations are of four types. Number 1, deficiency or deletion. Number 2, duplication. Number 3, inversion. And number 4 and last, translocation. Let us discuss these one by one. Thank you. Deficiency or deletion. It is loss of a segment of a chromosome which may occur due to several reasons. Just see in this example. You see green barred segment of a long chromosome has been lost and it has become smaller. Deficiency or deletion may be of two kinds. Either a terminal segment may get lost or an intercalary segment may get lost. The latter is called interstitial deletion. As far as deficiency is concerned, it is always deleterious and often lethal. In case of a deletion heterozygote, normal chromosome would form a loop to get extra genes away from the aberrant deleted homologue. Go to see the sketch. In case of both duplication and deficiency, during meiosis, when the homologous chromosomes pair, there is the formation of loop. In case of deficiency, the normal chromosome will form a loop, what you see in the diagram. And in case of duplication, the duplicated chromosome will form the loop. Formation of loop is because of the chromosome has to hide out those genes or parts of the chromosome which are not present on the second chromosome. Duplication usually causes accentuation of few genetic traits as if by polyploidy. What is inversion? Inversion is when an intercalary segment of a chromosome breaks but rejoins by rotating to 180 degrees at the same position. Thus, a genetic region gets inverted. If centromere falls in it, it is pericentric. If centromere is or this diagram shows you both types of inversions which take place in chromosomes. You see, on the left is paracentric inversion, when the centromere is outside the inverted portion, and on the right is pericentric inversion, when centromere is included within the inverted portion. What is translocation? It is interchange of segments equal or unequal by two non-homologous chromosomes. Just look here how translocation does take place between two non-homologous chromosomes. Heterozygous conditions of both inversion and translocation lead to very unique meiosis. They are respectively called inversion heterozygote and translocation heterozygote. The translocation heterozygote is also called interchange heterozygote. The inversion heterozygotes are called crossing over suppressors. Q, because they suppress the production of crossovers during meiosis. Let us see why. It is the case of pericentric inversion heterozygote. In meiosis, the pair of chromosomes form a special kind of loop called inversion loop. Look attentively. If a single crossing over occurs between the inner two non-sister chromatids, segregation in anaphase will form only parental types of segregants. Crossover will not survive due to duplication and deficiency. Just look deeply. Now it is the case of paracentric inversion heterozygote. See here, if one crossing over occurs, one crossover product will be acentric and the other will be dicentric. During anaphase, both will be lost. Dicentric chromosome forms a bridge between two poles of the dividing cell and breaks. Again here, parental types of meiotic products can only survive. Now we are looking into translocation heterozygote. Here also behavior of chromosomes will greatly be specialized. Due to partial homology between two pairs of chromosomes which have translocated with each other, multivalents of chromosomes are formed during meiosis. If two non-homologous chromosomes have been subjected to interchange, there will be a quadrivalent of chromosomes at metaphase 1. Such pairing will lead to a ring of chromosomes at late metaphase 1. Segregation during anaphase 1 might be adjacent or alternate. See the figure alternately. Just think which segregation will form viable gametes. Thank you.